Hello students, today we are going to study a very important topic known as the chromosome. These chromosomes are basically the hereditary vehicles as they are carrying the genes from one generation to another generation. To study the chromosome in details, we have to study the size, shape, number and the morphology of the chromosome. Very first, there was a scientist whose name was Strasburger, who have discovered the chromosomes during the cell division. So the credit for the discovery of the chromosome goes to Strasburger. And you must know that what is the meaning of the word chromosome. Chromosome is basically colored body. Chromo means colored, so means body. It is called as the colored body. First of all, this term chromosome was given by a scientist whose name was Waldier. These structures can be stained. If you want to view the chromosomes in the microscope, then you have to stain the chromosome, and the staining of the chromosome can be done with the help of basic fuchsin or the acetocarmine. Now very first, we are going to study the size of the chromosome. You must know that what is the size of the chromosome. Size of the chromosome varies greatly. It is actually from 0.25 micron to 30 microns. But before discussing the size, you must know that always the plants are having the more bigger chromosomes in comparison to the animals. You can see here the sizes 0.25 micron, which is called as the smallest chromosome, and it is found in the birds and the fungi. The birds and the fungi are having the size of 0.25 micron, but the greatest size, that is known as the 30 micron, this 30 micron chromosomes are found in the plant known as the trillium. If we talk about the human beings are having the size of 5 microns, while the Drosophila, we can say it as the fruit fly, is having the size of the 3 micron. So the size varies from 0.25 micron to 30 micron. But there are few exceptions known as the giant chromosomes, say for the polytene chromosomes, the lamp brush chromosome. They are having more sizes, approximately from 800 micron to 2000 microns. They are called as giant microns. But normally the size of the chromosomes varies between 0.25 micron to 30 micron. And if we want to study the size of the chromosome, the best stage of the cell division is the metaphase stage. And now see the shape. If we want to study the shape of the chromosome, the best stage is the anaphase stage of the mitosis. So shape can be studied at the anaphase stage and size can be studied at the metaphase stage. Now shape depends on the position of the centromere. The shape of the chromosome depends on the position of the centromere. Okay, now what is the position of the centromere? You can see that if the centromere is present at the tip, then the chromosome is called as the telocentric chromosome. This is a rod shaped chromosome. But see the acrocentric chromosome. In acrocentric chromosome, the centromere is present near the end. See here, in telocentric, the centromere was present at the end, but in acrocentric chromosome, the centromere is present near the end, and that's why it is called as an acrocentric chromosome. What about the metacentric chromosome, submetacentric chromosome? In submetacentric chromosome, the centromere is present little away from the center, and it is called as submetacentric. But in the metacentric chromosome, the centromere is present at the center, and that is because of which the two arms of the chromosomes are equivalent. Here the shape of the chromosome is V-shaped, while in the case of the submetacentric chromosome, the shape is J or L-shape. So the position of the centromere decides the shape of the chromosome. On the basis of the position of the centromere, the chromosomes are of 4 types: telocentric, acrocentric, submetacentric, metacentric. Here the centromere is at the end, here it is near the end, here it is present little away from the center, and here it is present at the center. Now view these two chromosomes. Now see here, this is a metaphasic chromosome. If you see the metaphase stage of the mitosis, the chromosomes are visible in this way. Here there are two chromatids, and at the center what is present? The centromere. The two chromatids are attached at a point known as the centromere. So this entire structure is a metaphasic chromosome. But if I isolate one chromatid, from here, then this is a single chromatid. What happens that in the anaphase stage, this centromere divides. So with each centromere, one chromatid moves. So this is a single chromatid. Okay, so anaphase chromosome is like this type. It has a single chromatid, while this is a metaphasic chromosome having the two chromatids. All right, so this was about the size and the shape. Now we have to discuss two more points. We have to discuss the morphology, as well as we have to discuss the number of the chromosomes. See here. Now the third point, that is the number. Number is constant in a species but variable in different species. The members of the same species are having the same number of the chromosomes, while the chromosome number is different in the different species. Now we must know least chromosome number in animals. We must know that the least chromosome number in the case of the animals is Ascaris. Ascaris is having the chromosome number of 2n is equal to 2. So in the entire animal kingdom, Ascaris is having the least chromosome number, that is 2n is equal to 2. But if we talk about the plants, the least chromosome number is found in Mucor himalis. Mucor himalis is having 2n is equal to 2, while Haplopeppus gracilis is having the chromosome number of 2n is equal to 4. Okay, so these are the least numbers found in the plants, these are the least numbers found in the animals. And if we talk about the highest number of chromosomes, in plants, so in plants the highest number is found in Ophioglossum reticulatum. It is consisting of 2n is equal to 1260, and in the case of the animals, the highest number of the chromosome is found in Olocantha. Olocantha is a protozoan. In Olocantha, the number of the chromosome is 2n is equal to 1600. Okay, so we know that number of chromosome is constant in a species, but it varies from species to species, and at least you must remember the least number of the chromosome in plants and animals, and you must know the highest number of the chromosome in plants as well as animals. Okay, and one more thing, that organisms have constant number of chromosomes generation to generation because of a cell division known as meiosis. Say our grandfather was having 2n is equal to 46 chromosome, our father were also bearing the 2n is equal to 46 chromosome, and we are also bearing the 46 chromosome. So the organisms are having constant number of chromosome generation to generation. The chromosome number remains constant generation after generation because of a cell division type, and that cell division type is known as meiosis. So student, this was about the number. We have discussed the size, we have discussed the shape, right? We have discussed the number, but at the last, very important thing which must be discussed, and that is the structure or the morphology of the chromosome. You can see the diagram I have made here to explain the structure of the chromosome. It's very important. See here. structure. We are going to study structure. You can also say that we are going to study the morphology. One by one, we'll be trying to discuss the parts of the chromosome. This is a single chromatid. This is a single chromatid. Okay. Now, first one, telomere. This is actually the tip or end of chromosome. The ends of the chromosomes are called as the telomere. Okay. And because of telomere, chromosomes are not united with each other. Getting there are many chromosomes in an organism. Okay, so the two chromosomes are not united with each another. They are not stuck with each another because of the presence of the telomere. So this is the chief function. This is the chief function of the telomere. And you must know that this telomere contains a nitrogen base known as guanine. It contains a nitrogen base known as the guanine. And this telomere, very important, telomere is associated with the aging in organisms. The major region for the aging is the telomere. What happens that as the lifespan increases, what happens? The size of the telomere become short. Okay, with the growing age, the size of the telomere is reducing. Okay, so we can say that telomeres are associated with the aging in organism. Aging occur because of the reduction of the size in the telomere. As our lifespan increases, what happens? The size of the telomere shorts and the person become aged. Okay, so these are the two functions of the telomere. The tip of the chromosome is called as the telomere. Now, the second one, this one, just concentrate here, that is known as the pellicle. Second part, pellicle. What is pellicle? So first of all, you must know that it is non-genetic. It is non-genetic part of the chromosome. Means it is not associated with heredity. Basically, chromosomes are associated with heredity. They are moving from one generation to another generation. But this part of the chromosome is basically non-genetic and it is also achromatic. Right? It is achromatic and it is non-genetic. Basically, pellicle is the membrane. Pellicle is the membrane of chromosome. The chromosome is surrounded by a membrane which is made up of the protein, and this membranous sheet found around the chromosome is called as the pellicle, which is made up of protein. It is non-genetic, it is achromatic, and it is not moving from one generation to another generation. It is not related with at all with the heredity. Now see here, third point, you can see this one, that is the matrix. Again the same thing, matrix is also non-genetic and it is also achromatic part of the chromosome, and this is the ground substance. This is the ground substance present inside the chromosome. Actually, this ground substance, which is gel-like, it consists of the enzymes. It consists of minerals, proteins, etc. But it is also not associated with heredity. So the pellicle part, which is the membrane, and the ground substance which is filled in the chromosome, both these parts are not associated with the heredity. They are non-genetic and they are achromatic. But one thing to be noted here is that they appear at metaphase stage when nucleolus disappear. Very important to note here that what happens when we study the cell division. So there are different stages: prophase, metaphase, anaphase and telophase. Now at the end of the prophase, the nucleolus disappear. So after the prophase, which stage comes? Metaphase. So in the metaphase stage, we know very well that nucleolus is not found. So when nucleolus disappear now, then at that time pellicle and matrix appear. But what happens when the nucleolus will again reappear? When the nucleolus will reappear, so what will happen? The pellicle and the matrix will disappear. So when vice versa, when nucleolus disappear, they appear, pellicle and matrix, and when nucleolus reappear, so they disappear, pellicle and matrix. So this was the third part, pellicle, matrix. Yet we have discussed the telomere, the matrix and the pellicle. Now coming to the fourth part, which is the most important part of the chromosome, and that is the chromonimeta. This one, this is the coiled thread like structure. Chromonometas are coiled thread like structures. These are too much coiled thread like structures found in the chromosome. Many people are having the confusion regarding the chromonimeta. So you have to clear the concept about the chromonimeta, that what is basically the chromonimeta. It is the part associated with heredity. This is the major part which is dealing with the heredity. Chromonimeta is basically made up of what? It is basically made up of DNA and protein. It is made up of DNA and protein, right? And we know very well DNA is the genetic material, it is associated with heredity. So literally the chromonimeta is associated with the DNA and protein. What we know when we study about the chromosome, if somebody asks you that what is chromosome, so chromosome is DNA plus protein. Chromosome is DNA plus protein. But now the question arises that when we study the morphology of the chromosome, where is the DNA and where is the protein? So this creates a confusion. Basically chromonimeta is nothing, it is associated with heredity, and when the chromosome become condensed, so what happens, that this condensed part is nothing, it is the DNA and the protein. So the chromonimeta is associated with heredity, and chromonimeta is coiled part, and it is made up of DNA and the protein. Okay, now coming to the next part of the chromosome, and that is known as the chromomere. What is chromomere? You can see that on the chromonimeta I have made various beads like structure. Okay, these chromomeres are bead like structure. These chromomeres are bead like structure, and they are formed due to condensation of DNA and protein. A more condensed chromosome have more chromomeres, right? So chromomeres are the bead like structures that are formed due to the condensation of the DNA and the proteins. A more condensed chromosome is having more chromomeres. Okay, one more thing I will be liking to discuss with you regarding the chromomere, that these chromomeres are gene bearing portions found on chromosome. It is said that the chromomeres are the gene bearing portions found on the chromosomes. Okay, so these chromosomes are actually, these chromomeres are present in the condensed chromosomes, right? And they are the gene bearing portions found on the chromosome. And at the last I will be liking to write here that note that chromonimeta are subunits of chromatids. The most important part of the chromatid is the chromonimeta, and on the chromonimeta certain granules are present, and these granules are called as what? The chromomeres, which are the gene bearing portions found on the chromosome. So this was about the chromomeres and the chromonimeta. Now next point, that is the primary constriction. This primary constriction is also called as centromere. Sometimes we also use the word kinetochore when we are discussing about the primary constriction. Now, what is kinetochore? Kinetochore are actually protein discs. There are two protein discs on the centromere, right? And what is the role of these protein discs? That during cell division, spindle fibers are attached to these discs, right? What I am saying, there are two protein discs found on the centromere region, and these protein discs are known as what? The kinetochore. During the cell division, the spindle fibers which are carrying the chromosome towards the opposite poles are actually attached to these protein discs, and this centromere is only called as what? Primary constriction. Why this part is called as primary constriction? Because this part is little bit shrinked part of the chromosome, right? It is little bit constricted part of the chromosome, and hence it is called as what? The primary constriction. Okay. Now, some chromosomes, not all chromosome, some chromosomes are also having another constriction, that is known as what? The secondary constriction. But remember that some chromosomes, only some chromosomes have the secondary constriction. Primary constriction is the chief character of each and every chromosome, but secondary constriction is not the chief character. Okay, only the few chromosomes have what? The secondary constriction, right? And the secondary constriction is surrounded by a part known as nucleolar organizer region. Very important part, nucleolar organizer region, right? In short, it is also called as NOR. You can see that I have made a secondary constriction here, but around the secondary constriction I have made a part known as the nucleolar organizer region, right? Why nucleolar organizer region? This part is very important because this part is actually having the RNA synthesis, rRNA synthesis. This part is concerned with ribosomal rRNA synthesis, means this part is responsible for the formation of the nucleolus. This part is responsible for the formation of the nucleolus, right? And here we can see that near the nucleolus organizer region, what is present? Nucleolus is present, and because nucleolus is present here, it is called as the nucleolus organizer region, right? And the last part of the chromosome, you can see that satellite, this part, right? A part beyond the secondary constriction, is called satellite or trabant. Basically, this part is pinched off. This part is pinched off due to formation of secondary constriction. Okay, due to the pinching of the chromosome, a part comes out, and that part is called as what? Satellite part. Okay, now what is the satellite? A chromosome bearing satellite. A chromosome bearing satellite is called set chromosome. A chromosome bearing satellite is called as what? Set chromosome. Now, what is the full form of the set? What do you mean by set? So, sine, acid, thymo, nucleo, nico. Okay, this is the full form of the set chromosome, right? Sine, acid, thymo, nucleo, nico. Okay, so this was the satellite. So we have discussed the morphology of the chromosome: telomere, matrix, pellicle, chromonimeta, chromomere, primary constriction, so called as centromere, secondary constriction surrounded by nucleolus organizer region having the nucleolus, and a part which is pinched off beyond the secondary constriction is called as satellite. A part of the chromosome which bearing satellite is called as the set chromosome. Set means sine, acid, thymo, nucleo, nico. Okay, so this was the morphology of the chromosome. We have discussed the size of the chromosome, shape of the chromosome, number of the chromosome, and the morphology of chromosome. few more points we have to discuss regarding the chromosome. If you have heard about the autosomes and the sex chromosomes, chromosomes are basically categorized into two. One are known as autosomes and another are known as sex chromosome. To explain that what are autosome and what are sex chromosome, I will be liking to take an example of human being. As we know very well that human beings are having 2n is equal to 46 chromosomes, means we are having 23 pairs of chromosomes. Okay, so out of the 23 pairs of the chromosome, the first 22 pairs of the chromosomes are basically what? Autosomes. And these autosomes are actually controlling secondary sexual characters. They are not related with the sex determination. They are only associated with the development of the secondary sexual characteristic. And remember that whether it is a male or a female, these autosomes are same in both male and female. But now the question arises that if the autosomes are controlling the secondary sexual characteristics and if they are same in male and female, then why they are the secondary sexual characteristics are different in male and female? So you will answer it that because of the impact of the hormones. Because of the impact of the hormones, the secondary sexual characteristics are different in male and female. Okay, but here sex chromosome, that is the 23rd pair of the chromosome, that is the last pair. If we talk about the human beings, the last pair, that is the 23rd pair of the chromosome, is the sex chromosome. Okay, and this sex chromosome is responsible for determination of sex. What is the chief role of the sex chromosome? That they are important for the determination of the sex. Whether the born child will be a male or a female, it wholly depends on the presence of the sex chromosome. Remember, XX is the sex chromosome found in always the female, and XY is the chromosome pair which is found in the male. Right? So this was one more classification of the chromosome, that chromosomes may be autosomes and sex chromosome. Now at the last, based on the staining, the chromosomes are classified into two. Okay, so based on staining, we can say that chromosome have either heterochromatin part and another is euchromatin part. Okay, this is the classification based on staining, heterochromatin and euchromatin. Okay, this heterochromatin is darkly stained part. This is darkly stained part and this is lightly stained part. Okay, when we stain with the acetocarmine, so the darkly stained part is the heterochromatin and the lightly stained part is the euchromatin. Okay, now this is genetically inactive part. This is genetically inactive part, but the euchromatin is genetically active part. Okay, now it do not participate in transcription. It participate in transcription because it is genetically active. It has less DNA. It has more DNA. Okay, so based on the staining, the chromosomes are of two types, heterochromatin and euchromatin, and this heterochromatin and euchromatin word were first of all given by a scientist known as Emil Hertz. There was a scientist known as Emil Hertz who gave the heterochromatin part and the euchromatin part. At the last, what is the chemical composition? If somebody asks you that what is the chemical composition of the chromosome, that chromosomes are made of what? So you must know that chromosomes are made of DNA. Okay, DNA. They are also having RNA. They are having non-histone protein. And they are also having the histone protein. Okay, so they are made of DNA, RNA, non-histone protein and histone protein. So students, this was the description of the chromosome. Okay, if you have liked this video, please subscribe my channel and also like and share this video.